Hi everyone. I wanted to do a little tutorial on using ChimeraX. ChimeraX is a program distributed by the University of California, San Francisco that visualizes 3D structures of proteins in general and other molecules as well. And so we're going to use that to create a series of figures to help guide our readers for our final report to knowing where you made the mutations. They can see where it is in relation to the active site and things like that. So the first thing to do is to go to the Chimera website, which is shown here on the screen right here. It's this cgl.ucsf.edu/ChimeraX. Once you get to that site, then go ahead and just click on the download link. And the download link will have, depending on your operating, it automatically detects your operating system. So it'll know that I have a Windows computer here. If you have a Mac, it'll come up with Mac. And I think if you are using a Linux computer, it'll come up with Linux as well. Go ahead and download that and get it going. And I'll assume you can do all that because it was a pretty easy setup. What I'll do is I'll minimize that screen. And if you start Chimera up, let's go ahead and do close session here. Now, I've played with a number of different molecules here over time. So whenever you first open it up, I don't think there'll be any files in this portion right in here. But here, what it is that we have this AlphaFold file here. We'll click on it, and we see there's an ion here in kind of this weird sort of cyan color and this sort of light blue kind of color for the rest of the molecule. So when you first open this up, if you hold the mouse wheel and kind of roll it towards you, you see the molecule get smaller. And if you push it away from you, it gets a little bit larger. If you go ahead and hold the mouse key around, you can rotate and move the molecule around. I think it centers on where you put the cursor. Let's go ahead and make it smaller so you can see the whole molecule. Here, if you hold down the wheel of your mouse, you can move the structure around this way. And just move it, rotate a little bit this way, maybe a little bit this way, here. You know, you look around here and it looks like, oh, this looks like this is probably the active site of the enzyme. So let's go ahead and leave that right here. Make it a little bit bigger. Let's move it a little bit over. So get it centered in the picture here. And we'll start working on our first illustration. What I'm thinking is, I think having the structure in a neutral color like gray is a good idea. Let's go to Select, Chains, and this 4-nitrophenyl phosphatase, and it highlights everything except for that ion right there. Let's go ahead and go to Actions, Color, and make it light gray. So now we have the thing in light gray. Now if you want to go ahead and unselect what's been selected, go to Select and Clear. So now we have this gray protein structure in the cartoon form. When you see these little arrows for beta sheets and helices like this for alpha helices, you know this is referred to as a cartoon version. There are space filling modules like that you may have seen in lecture class, but here it is. Let's go ahead and let's change that ion here. And to select that, you go to Select, Residues, and this CA. It's selected. Go ahead and change it to red. So go to Actions, Color, and Red. Now I'll go ahead and deselect it. So now we have it in this color. And so I'm looking around, and the active site is here. And to go ahead and show you what you do for the rest of the assignment, I may as well go ahead, and the earlier examples this year was, for assignment 2, I gave you the example of everything for the mutation of the alanine 72 being changed to valine, referred to as E72V. So let's go ahead and select that. Well, to be able to select amino acids, the easy way is to have a sequence pop up. And to do that, let's go ahead and select the whole chain again. Go to Molecule Display, which is the second row of tabs here, which is kind of a little bit confusing, but once you get used to it, it's not too difficult. And click Sequence. And here you'll see the sequence appears in the lower right-hand corner. So Let's go ahead and look at A72. So you see this is A61 here. So that's 61, 62, 63, 64. And so the A72 is here. And to select an amino acid is a little bit interesting. You have to kind of swipe across it. So you have to drag across it to select it. At first it seems a little bit awkward, a little bit strange, but after you get used to it, it isn't too difficult. So we'll select that. And let's look around. And if you look here, it's kind of hard to see, but there's a light green line all around this area. So this is where alanine 72 is. Let's do a few things to make it maybe a little more obvious. Let's go back to the Home tab. Click on stick. See, this residue is, this alanine is still selected. Click stick and then show. And you'll see that the methyl group pops up right here. So we'll go ahead now. We still have it selected. We'll go to Actions, Color, and green is a nice bold color. So we'll go ahead and have it in green. So we have this color in green. I go because I don't like the idea of seeing the little outline on it. So I'll go ahead and I'll deselect it. Click Clear. Now, I want to go ahead and capture a picture because we're going to create an illustration showing where this residue is in relation to the active site. You see the active site is this area right in here. And we'll go ahead and a little bit later, go ahead and zoom in on that active site. But this time let's go to the broad view first. Go to File and to Save. Chimera is very powerful. It lets you save lots of different formats. And here currently the format would be a ChimeraX session. I don't like that. I want to make a JPEG so I can easily put it into Word for my report. And I'll scroll down here and there's JPEG. I think there's, if you want, there's a PNG as well, JPEG. If you like GIFs, there's a GIF. I kind of JPEG is easy to work with. We know that all programs will accept that, so just go click on JPEG. This will just let you know a little bit of information here. This is the size the picture will be. It'll be 948 pixels by 592 pixels. That seems about fine. So what I'll do here is type a name, A72, A72 in green, zoomed out. We'll click Save. So now we have a picture. You can see over here, you can verify what it did, and it tells you what it did right over here. Now, we want to go ahead and change this amino acid to a valine. And we can do that in this way. So let's go ahead and select that A again. See, now it's all selected. The light green line is all around it again. We will go to right mouse. And there's a button over here called Swap AA, Swap Amino Acid. So we'll go ahead and click on that. Take your cursor and go over the amino acid you're interested in changing. Right click on it and drag down. And you'll see all these little possibilities start to pop up. Keep going, 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 going. Oh, come on now, go here, keep going down. Your serine, threonine, tryptophan, tyrosine, there's valine. So now we have a valine here. What I'll do is, if you notice, the light green line isn't around the sticks for the valine. So what I'm going to go ahead and do is I'm going to reselect that valine. So it's now completely selected. A nice little thing to do to help your reader out is maybe label that amino acid. So let's go ahead and we can, and Chimera will let us label that amino acid. So what we'll do is here we'll go to Actions, Label, Residues, because remember, amino acids are referred to as being residues. Let's go ahead and put name and number. That'll be a 3-letter abbreviation for the name and the sequence number that it is. We'll click on that. And you see it's in really small print right here. That's not that useful to me. So what we'll do is we'll go to Actions again, Label, and then Set Label Height. And it has it down as 0.7. Let's make it 3. I think 3 looks good. Let's see. That's maybe a little bit big. Let's make it 2. There it is. So it's Valine 72. We'll click OK. Now let's go ahead and save this picture. Oh, let's go ahead and deselect again. Clear. File. Save. We'll do A72V. We don't want to do that yet. We want to go ahead and select that amino acid again. And we don't want it to be the same color as the wild type. So let's go ahead and change the color. Go to Actions, Color, and we'll make it cyan. So it'll be a contrasting color. So you see the two little illustrations. You'll know each of them are different, because one of them was a change, and you'll know the green one. And you can put that in your figure legend. So let's go ahead and deselect. Go to File, Save. A72V labeled, zoomed out. Okay. Now we have two pictures. We're going to have to go back potentially and change the A72V and label that A72, but we'll go ahead and I'll show you a way to go backwards and do that later. At this point, let's go ahead and do it now. I think it'll be one step easier. Let's go ahead and select that letter. Do swap amino acid. Right click. Kind of go up. There's Alice 72. Let's go ahead and Actions, Color, make it forest green. And let's go ahead and save that file. Notice here it says JPEG again. So we'll go A72 green labeled and zoomed out. So you see how easy it is to save the picture. So you can save a whole bunch of pictures and then later on decide which ones fit your report the best. So we've got this in place. Let's go ahead and select that again. Let's go ahead in Actions, we'll make it color and make it forest green. It's made a whole thing green. So we'll do that. Now let's go ahead and zoom in on this area here. The active site is this area right in here. This is the mutation we have. Let's go ahead and make that area bigger. Move it around. Let's go ahead and zoom in. Let's rotate this around so we can see maybe in space here. The active site looks like it's in this area, probably right in here. Here's the A72. So this looks like a nice arrangement. So let's go ahead and do this. Go to File, Save. We'll call it A72 in green. Labeled and zoomed in. Click Save. So we have this A72. We have it all nice and labeled. There's the end. There's the whole active site. So this might be a really good way to explain kind of what's going on and where this amino acid is in that site. So maybe there'd be a specificity change or something like that. So we have that. Now let's go ahead and make sure that amino acid is selected. I always go ahead and it's so easy to select them, I reselect it. Let's go to right mouse, swap amino acid, right click, and I'll drag up here and we'll come up, hopefully, there it is, valine 72. Now we have the valine here. We saved a picture with the A72 close there, and this is where the valine is. So we'll go ahead and type that do this one, do save, A72V. We didn't, I forgot again. Let's go ahead and change the color. Actions, color, we'll make it cyan to make it correspond to the last one. We will deselect and go to File, Save. And it'll be A72V in cyan, labeled and zoomed in. Click Save. So what we've done is we've now saved 4 pictures. So we have A72 in green in the zoomed out version and in the zoomed in version. We have the mutant A72V zoomed out and zoomed in. So we have 4 pictures showing the active site, and you can go ahead and put all 4 of those into a Word document, create a figure legend for each one of them, and you turn that in for assignment 4. And that's a nice little document for you to be able to use for your final report that you'll be turning in later in the semester. Thank you. If you have any questions, feel free to contact me and we can walk through it. To let you know, if you want to use, if you have trouble running Chimera on your computer, if you want to run it, the lab computer has it as well, and you can come in and I'll let you sign in on that computer and be able to generate your drawings. I will see you later. Good luck. We'll see you later. Bye.